So the project for this module is our fish dissection. And there are two options for those of you that get a little queasy with dissections. You can also turn it into an art project. So we'll talk more about the details of the project once we do a little bit of studying and reminding ourselves of what zooming into structures within organisms actually looks like. That topic for topic 3. So I wanted to start off just with a bit of a discussion. And if you open up the document that's in the agenda called the lesson for today, you can see that there's a Venn diagram right at the top. And we love Venn diagrams. But this Venn diagram is specifically showing us what is similar and what is different between invertebrates and vertebrates. So I remember when we started off this course, we were learning a lot about plants, and we were learning about vascular plants, and we were dividing up plants into all these different structures, and really getting to know the different parts of plants and the history of plants and reproduction, and we got pretty nerdy with that. We're kind of taking that same approach now as we dive into species. So into specifically animal species, the kingdom Animalia. So in this kingdom, we have both vertebrates and invertebrates. So based off of that word, what do you think vertebrate means?
With vertebrae.
With vertebrae, exactly. So species with that internal vertebrae, that backbone, right? And then invertebrates are exactly a spine as we know it, specifically in animals, this upright spine that we get to carry. But species without a backbone are called invertebrates. And we've talked a little bit about invertebrates. I mean, insects, different types of worms. So all of these things are, and you can see on the left-hand side, now we're starting to read this list, all of these things, of course, have no backbone. They're multicellular. No cell walls. They actually reproduce sexually still. They are usually smaller, slower moving. The quantity of these species is huge, even though they are tiny. I know we've all read crazy statistics about, if you piled up the number of ants in the world. What's that statistic? I can't remember it off the top of my head. It covers something like greater than North America or something.
I have no idea.
Yeah, it's crazy because there's so many species that are living underground, right? If you start to think of lots of these invertebrates, they're living actually within the soil. So they're not above ground. So huge quantities, huge numbers of them. We don't talk too much about the invertebrates within the Biology 11 course. We really focus more on vertebrates when we're actually looking at the study of animals. So in this topic, we've decided to focus specifically on fish. Which is interesting because I think a lot of people don't really think about the fact that fish are still vertebrates, and anyone that's ever eaten a fish, even if it's been mostly well taken care of and cleaned, often there is still that backbone, right? You guys have noticed that, and that's where all the smaller bones come off of. So if we look on the right-hand side of our Venn diagram, then we can start to look at some of the qualities of the vertebrates. So things that make them different than the invertebrates is that they, of course, have that skeleton, that internal structure or that backbone that we know. They've got a really well-developed internal skeleton as well. So there's, lots of bones, not just a backbone. More highly developed and evolved in terms of, the functionality. They actually do have a very developed brain, advanced nervous systems, advanced all of the systems actually, the vascular, lymphatic, cellular. A few examples, of course, humans are vertebrates, but, birds, even snakes, which is really cool.
They have so many bones.
So many bones, right? You don't think about that, though, when they're moving around. And these vertebrates are classified into 5 major groups. So fish, amphibians, reptiles, birds, and mammals. You guys learned that in something like grade 3 biology, and often larger in size. So the interesting thing about the 2 of them in the center of the Venn diagram, you see that what they share is that both vertebrates and invertebrates are part of the Animalia, or the animal kingdom. And they are both in the same phylum. So we start to look at these levels of classification. If you guys go down one level here, or one image, and we talked about this really briefly last week, but I wanted to bring it in again and start to give a little bit more information about the levels of classification and where this even came from. So we talked about the 7 levels of classification, going from really general, right, that large kingdom, down through kingdom, phylum, class, order, family, genus, species. So we're actually getting more and more specific as we get lower down in these classification levels. The biggest branches, we talked about there being, I said 6, but lots of people just say 5 because they clump bacteria and the archaebacteria together in one kingdom. So if you just remember that bacteria is usually 1, but you can divide it into 2 as well. And so we talked about the kingdoms last week, but we didn't talk about when we use the genus and the species. So the species is described by its unique name, which is a combination of the genus and the species, like Homo would be the genus, and sapiens would be the species. So the way that we write this, and you've all seen this, is in italics. The first word has a capital, and the second word has a lowercase letter. So that's important because you're going to run across that a lot. And even in your studies for classifying your specific fish that you decide to dissect or draw, I'll be asking you to figure out the classification of it.
So the first word is going to be the genus and the second word is the species?
Yeah, exactly.
And there's always 2 words?
Yeah, there's always a genus and a species, yeah.
Awesome.
When you're doing this type of classification. So the system that we use was developed by Carolus Linnaeus in the 1700s, who was the first person to actually create this this taxonomy, so this system of classification of species, and he did it through recognizing that there's different characteristics within species, and he was really basing this solely off of physical characteristics and looking at what is similar between species and clumping them together that way. So why do you think this was of huge benefit to biologists to start to look at physical similarities and clumping them into groups?
I guess because it's a way of organization, and sort of like if you find something new, then you can know that it's similar to that, so it can be considered a whatever, and then you can go deeper into its makeup. Yeah.
Definitely. And why else? What do you think you're learning if you're starting to learn about one species? Do you think it's going to share common characteristics as things that look similar to it?
Yeah.
Exactly. All the way from its the functionality of the species, the way that it, like the biomechanics, right? The way that it moves and breathes and eats and drinks, but also all the way down to the internal biomechanics of how it is designed if you're to dissect it, right? So what is the structure? What is the positioning of the bones? Why are they positioned that way? Start to look at quadrupeds, right? In the Mammalia, so in all of the mammals groupings of species, you can really see similarities if you start to look at ungulates, right? Species that have hooves. You see that they all carry similar functionality for why they developed hooves in the first place. And their biomechanics are very similar because of that. So it really, Linnaeus's classification system really actually saved us as biologists a whole lot of time. Being able to group things together. And then suddenly we're able to actually study not just an individual, but a collection. And start to hypothesize as to why these collections have adapted these certain traits. And what traits helped them to succeed and continue to reproduce. right? So now we're looking at Darwin's natural selection. So this is an amazing sort of overview of a lot of the scientific history that you guys have heard and studied a little bit in Science 10. Now you're starting to kind of piece this together, specifically in how it relates to biology. The other piece here is that sort of 2-word classification of species that Linnaeus came up with was, the word for that is binomial nomenclature. So that's the name of the species made up of 2 parts, or a 2-part name. Binomial nomenclature. You guys might hear that. Okay. I get excited about this stuff, so sometimes I rant. You guys with me? Okay.
Yeah, I can't hear very well, but I'm still here.
Okay, I'll talk a little closer to here.
No, it's like I can hear every second word because it's lagging.
Oh, dang.
I'm trying to listen.
Okay. Well, it is recorded if you have to go back to any of it. The last image, you guys, I gave a specific example for a fish of the scientific classification. And this is what you'll find if you even just look up in Wikipedia. So you'll be able to very easily classify your species of fish. And I'd love to kind of hear a little bit about, so in your project for you to do just a little bit of research into one part of the classification.
Into the what of the what? Sorry?
Into like one part of the classification. Like, we all know why it's in the kingdom Animalia, but maybe describing one of the other pieces. You know, why is it in the phylum Chordata? Or why is it in, the class Actinopterygii? You know, or why is it in the order? So we start to get into all these Latin names that we would never use outside of this classification system. But it is important, and I want you to know sort of where that came from. So get curious about that when you're doing your own investigation for your project, okay?
Okay.
I'm not asking you to memorize all of them. You're lucky that we're not taking this in a regular high school, because there would be a whole lot more memorization.
No, I'm not upset about anything. It's just the connection is killing me.
Why don't you turn your video off?
Okay.
That'll use less bandwidth and help you out. So that's my little spiel for our intro. And go back to your agenda and go ahead and open up your project. So for this part, I am sharing my screen for anyone watching the recording. A little bit easier to follow. So this is our 5th activity, anatomical fish dissection. And here I describe that dissecting a fish is an excellent way to explore the structural and functional hierarchy of a vertebrate, or vertebrates rather, particularly its organ systems or its internal systems. Now, fish are readily available as whole specimens if you go to the seafood section of your grocery store. You might be surprised.
Are they going to have innards in them? Because you don't buy fish with the whole fish not gutted.
No, like,
how are we supposed to do this?
At some stores, you honestly can. So ask around.
I just wouldn't eat it afterwards then.
Wait, you wouldn't eat? Yeah,
I wouldn't.
I don't know if you're going to want to eat this fish because it's going to be out on your counter for quite a while you're playing with it.
Yeah, I know, but I'm just saying I wouldn't fish with the innards in it and then eat it. I don't think that's a great idea.
Yeah, well, it's not easy, that's for sure. So most people do go for a sort of gutted fish, but one that still has a backbone and skin and eyes and all of that. So this is definitely going to be an investigation to see what's available to you. And I mean, some of you guys live in places where maybe there are fishermen and you can go to a dock and actually pick up a fish. So there's that option as well. But I'm curious to see what you're able to come in contact with. And remember, this is the first time that I've run this lab as well. So it'll be good feedback for me to hear whether or not you were able to get a fish to dissect. But I thought it's kind of one of the easiest things for us to dissect. I mean, a fish is easier than a cow's eye, right? Or do you guys want to go get one of those?
No, I'm good.
That would be more physics and optics, though, so that wouldn't be as pertinent to Bio 11. Maybe a frog. We could try and track down a frog, but I think they'd be hard to...
You gotta go catch one.
I know. So I shared that the classic fish that you usually dissect is a perch. But obviously most fish do share quite similar internal anatomy.
Oh, well, there you go. You can catch one of those, Ashay.
I have no fish catching skills.
You don't need them.
Believe me.
Perch
I bet somebody you know does.
Maybe.
are so easy to catch.
Are they? See, I don't know too much about fishing either.
I don't know. You just go off any dock, and there should be like 1 billion of them right under you.
How big are they?
Okay.
Oh, wait.
I can't see
They're
you.
like, I know. Hang on.
Oh yeah, your screen's...
They're not that big. Like, you're lucky if you get one this big. Usually they're like this big or like anywhere. My friend caught one like this big one time.
Huh.
They're pretty small.
Wow. Are you going to try and catch one for us then, Bryn?
Oh yeah, I'm definitely gonna catch my own fish.
What? Awesome.
It's gonna be great because then I can go out for a day and not be like, well, I'm not doing school, because I am doing school. So it's gonna make me really happy.
See, I love you make
That's so
projects, Sarah, that make us do the things we love and become good at school. We can
Well,
run
it's a
around in the forest
challenge.
picking mushrooms and be doing schoolwork.
Yeah. I know Halima, though. Like, Halima's right in the city, right, Halima?
Yeah, I am.
So it might be more challenging for you. But, I mean, but you're also, in the city, you also have the benefit of having, like, really amazing stores, right? Like, not just having, like, these guys on the islands have, like, one grocery store, probably.
Oh.
Like, you have access to so much. Like, I bet you'll be able to find a fish.
I know, I'm going fish searching tomorrow in town. This is the
Okay.
I might do the art version because I don't know about opening up a fish.
The dissection. Well,
Yeah.
at least Bryn will be doing the dissection, and I think a few others. So at least we'll be able to, when we do our sharing for this exercise, you'll still be able to see some pictures of it.
Okay.
Anna's coming
Because
in.
oh, yay. It is, yeah, it is really cool. Okay, so then I also share that you'll have to use a sharp knife or a short blade. There's all sorts of different dissection tools. If you have a fisherman in your family, they'll, I'm sure, have some sort of really awesome knife you can use, but anything will do. And you'll make 2 lengthwise cuts, exposing open the digestive system, reproductive system, and the heart of the fish.
What do you mean?
Anatomy,
lengthwise
fish.
cuts? Like on either side
Fish
of the
and-
fish?
Yeah,
Okay, got it.
yeah, just to open it fully up. And the anatomy of the fish it's so easy because everything actually really is laid out. It's not hidden the way, you know, dissecting more complicated species is. So I think that you guys will actually find that this is pretty simple. And I haven't really included many resources because I know it's going to be different for each of you. Some of you might utilize someone in your family that you know is, you know, already has that information. You could definitely utilize the internet. Maybe you go to your library and pick up a biology of fish book. So there's all sorts of research out there. Go ahead, Bryn.
basically, if you want to open up a fish, you just stick the knife up its butt, and then you go all the way to its neck, and then there it is. It's
Can you
open.
cut the organs?
All it's in.
Did you say, can you cook them?
Won't
The
you cut
what?
the organs?
She's asking if you can cook fish organs.
No. Won't you cut the organs?
Oh, I think you said cook. I'm like,
Sorry, you're really super quiet. I can't really hear.
She's asking if you'll cut the organs when you do that.
You only have. You should you will if you stick it all the way in. You should stick it in as little as you possibly can. The knife.
Oh, okay.
So just go.
And then you can use your hands to manipulate and kind of peel it open a little bit too.
Yeah.
Ellie
So
really don't
usually-
look excited about this.
Usually when I do this, I do make one smaller cut first, and then I start to peel it open, and I use my knife to kind of go around different organs that I want to keep intact. Yeah. So, again, we're going to be writing this up like a lab report. So make sure you include the headings, objective, methods, results, discussion, references. Is Anna still trying to get in?
She's trying to get in. She says she's on a phone, and so for some reason it's not working.
It doesn't work very well at all.
On phones?
No, it doesn't.
I've never tried.
It's not fun.
She's going to keep trying though. Okay. Well, we are recording it for her. Remind her that.
What?
And,
Ranesh just popped into our chat. Weird.
Oh, interesting. Yeah, he didn't want to be in this course anymore. It was too much for him. Just because he's doing a lot of other school at the same time. So objective, be specific to your own assignment, depending on whether you're doing the art or you're doing the dissection, and then sort of what your own make it personal, why, you're in a biology 11 course and I'm making you do this, but also what do you want to get out of this? Or what are you curious about? The methods, so you're going to include obviously safety, equipment, collection location, and the different methods that you're going to use to actually dissect the fish. Images of the equipment or fish prior to this dissection would be useful. Again, I like that for the authentication purposes, just to show me that you actually did it. And then I would love a few images, if you are doing the dissection, a few images of your fish open, labeled, and I think it even says here in the discussion, state the identity of the organ and its role in keeping the fish alive. So in your results, you're actually going through, I didn't include the video, I'll add that in afterwards. They're going to be presented as a series of images or a video, if you wanted to make a little video of you opening up the fish, through the various stages of the dissection. And in the references, we're not using the Pinterest board, but I basically already mentioned that you're going to basically use your own references. The assessment's really helpful here because I actually outline in a little bit more details what I'm expecting of you guys. So in the results, I actually specifically want to see an image or images of external anatomy that include the labels with the arrows or close-ups of at least 4 structures. And then images of the internal anatomy, including the labels with clear close-ups or images of removed organs. So you can either take it within or take them out, with captions or labels for the pictures of at least 5 organs. So I wrote them down for you. Other structures to look at could include, and then you have a whole bunch of other ones. But of course, depending on what resource you're using and what ones you're actually able to find and really successfully label, you are free to include whichever other 10 ones you want, right? Those main 5 ones I want to see, and then the other 10 are up to you.
So you want a total of 15.
Exactly. Okay. Then in the discussion, at least 9 functions in total of the structures and organs identified in the results. So at least 9 out of the 15, you're going to go through and say what the function is, what was its role in keeping the fish alive, And then you get marks for whether your write-up is neatly presented or there's blood splattered everywhere. Just to make a little joke. At the bottom, it does say note, you can definitely create an art project using paper and other materials that shows both the external anatomy, so the body parts of the fish, and each of the internal organs and where they're located. You would also still want to do, follow the lab write-up if you're doing the art project.
I really want to dissect a real fish, but I also really want to just make a paper mache fish with all of it.
Whoa, that would be pretty cool.
Just do both options because I could. Although that's a horrible idea.
You have lots of time. We have...
Maybe if I have extra time over the holidays, I'll make myself a paper mache fish.
Yeah, because remember that at the end, we only have one more project that we're going to be doing. Depending on how this one goes, I think we'll probably start our other project after the holidays. And then the last 2 or 3 weeks of the course, you're actually going back into one of the projects that you loved and embellishing it, right? And doing it, so you're creating more of a unique experience for your Biology 11 course by going back into something and being I remember I told you about that when the mushrooms, if you collected more mushrooms, or herbarium if you collected more things. Or if you're like Ashe and you're interested in this, maybe you're doing both the art project and the dissection. So that's something that you could be including in your final part of our Biology 11 course. And that goes into your final portfolio for me. How does that all sound?
Sounds good.
A pretty cool, cool project.
Yeah, that doesn't sound too bad.
Nice. So next week, I actually want to meet with each of you one-on-one and just chat a little bit about assessment and just give you guys a really good idea of where you're at in the course with your marks and give you some feedback on some stuff that you've done and just a check-in. I think it's important for us to do that now. So next week, it's either on Wednesday or Thursday that you and I will have a one-on-one for Biology 11. And then we'll also do another one of those at the very end of the course, so probably the last week of January or something. So if you guys go back into the agenda, and you can plug, the 3 of you that are here, you can plug your name beside one of the times that I listed. And then I will send out an email to everyone else and see when they want to fit in. So you can just put brackets after the time and put your name there. Yeah. Perfect. So Ashe, 9:30 on Wednesday, Halima, 11:30 on Wednesday, and Bryn, 10 on Wednesday. Awesome. Cool. That's fantastic. And then I did want to just mention to you guys, because I know you're a curious bunch. Our last project in the course is looking at the 4th topic of kind of zooming, it actually covers a whole bunch of different big ideas, this last one. But there are going to be 2 options for the last one. One of them, depending on where you live, is a snow study in a snow lab. And then the other option is the ornithology study, so the bird feeder lab. The 3rd option I have is an aquatic insect lab, but that one would happen if we would have had to do this project earlier on in the year or later on in the year. So I'm thinking that we'll probably just have the 2 options, either a snow study lab or a bird feeder lab. And I will try to provide you with that information before the holidays. Again, in case you guys are really up to speed and you're really loving this course and wanting to be outside lots over the holidays and, can kill 2 birds with 1 stone, for lack of a better term, and do a little bit of biology if you want. So again, I'll try and have both of those ready for you guys next week, and we'll send out an email with all that information and where it is in Mahara. And then, yeah, the last month of our course is super laid back. We're working on some presentations, so sharing what we did for the fish dissection, sharing what we're doing for the winter lab option, and then working on that more specific kind of zoom into one of the projects that you were really excited about and felt like you didn't quite have enough time to do, and you get to go back into that and kind of embellish it. And then we do a big wrap up as a group. So there's no surprises. There's no tests, quizzes, exams. It's a pretty playful end of the session. We do a little mini exit interview, which is, a one-on-one reflection where you guys kind of walk me through some of the stuff you did in Mahara. And that's it. So just wanted to share that with you because I know it's a busy time of year and you're probably feeling stressed in some of your other courses right now as you go into the holidays. So hopefully you're feeling on top of this one, or at least like it's manageable to be on top of.
This course is the sanity in my school life.
Well, that's nice. Lots of people don't feel that way about biology, so that's awesome.
How come? What's wrong with them?
Awesome.
It's the easiest course and the funnest course out of all of them.
Yeah, that's true.
No, it's super easy because you don't even have to keep yourself on track. You keep us all on track for us. And we just have to do what you say
It's the
each week.
best.
Perfect.
Much sense.
When is this due?
Yeah, so good question. I think that we'll do our presentation for this when we get back from the holidays.
Okay.
Because next week we're just meeting one-on-one. We're not meeting as a group. So, yeah, we'll do presentations the Wednesday that we get back from the holidays. How about?
Yeah, that works.
Okay.
Yes.
Perfect. Well, I'm going to stop recording for those watching because I like to keep these recordings short. So thanks for watching if you were.